My name is CJ Barnum, Vice President of Neuroscience at AAIC Bio. Today I will talk to you about the results of the Phase 2 study that we reported at the Alzheimer's Association meeting this past week. These are the forward-looking statements. This Phase 2, 6-month randomized placebo-controlled trial evaluated the impact of XPro in early Alzheimer's patients with biomarkers of inflammation. Early AD patients, those with mild cognitive impairment or mild Alzheimer's disease, that were both amyloid positive and had at least one inflammatory biomarker as described in the key enrollment criterion box, were eligible to participate. 208 patients were enrolled into the study, and the primary endpoint was the early mild Alzheimer's cognitive composite. Key secondary endpoints of cognition, function, and biomarkers were also measured, and some of those will be discussed today. The baseline demographics are relatively balanced across the two treatment groups. The mean age in this study was 72.5 years old, with females representing roughly half of the study population. MCI accounted for 45% of the patients, while 54% of the patients had mild AD. The XPro treated group was a bit more advanced based on CDR, with a baseline score of 3.23 compared to 2.67 in the placebo group. 58% of the patients had at least one APOE4 allele, and most patients had two or more biomarkers of inflammation. As we have described previously, the primary endpoint was not met in the 6-month period. And looking closer at the data, the placebo group did not decline despite enriching for biomarkers of inflammation. It was only when we looked at patients that had high inflammatory burden and were also amyloid positive in our pre-specified sub-analyses that we observed a decline in placebo-treated patients. To evaluate a subgroup after missing the primary endpoint, we used effect size as the primary metric due to the small sample size. Effect size, measured by Cohen's d, is well suited for small samples and allows comparisons across different measures. For example, cognitive tests and biomarkers. Unlike p-values, which indicate the likelihood of results being due to chance, effect size reflects clinical relevance and is commonly used for signal detection in Phase 2 studies. We defined a promising signal as a minimum effect size of 0.2, where XPro outperformed placebo on multiple endpoints aligned with our hypothesis and the drug's mechanism of action. Results must also be appropriate for the trial's parameters, meaning the observed effects should align with the trial's duration and endpoints. For example, if a clinical measure typically requires longer time to show meaningful change than the trial's 6-month timeframe, an observed effect on that endpoint would not be considered supportive. Signal detection was based on the effect size difference in least square mean change from baseline between XPro and placebo at 6 months, ensuring results were meaningful, relevant, and appropriate for the trial's design and objectives. Pre-specified sub-analyses in the enriched population showed trends toward improvement with XPro on the primary endpoint EMAC and a key secondary endpoint, the neuropsychiatric inventory. With the placebo group showing the expected decline on EMAC over 6 months, a beneficial effect of XPro became evident. EMAC, which measures cognition, showed an effect size of 0.27, exceeding the company's threshold of 0.2, though the p-value fell short of the 0.1 target. For neuropsychiatric symptoms, the enriched population showed a stronger beneficial effect compared to the overall population, with an effect size of 0.23. There was no effect on CDR, which measures both cognition and function. Finally, this forest plot shows the effect size of the endpoints that have currently been assessed. These effect sizes, though small, consistently favored XPro over placebo on the primary endpoint and multiple secondary and exploratory endpoints. Effect sizes are particularly useful for comparing drug effects across different endpoints, especially with a smaller than expected sample size. Across most endpoints, XPro showed favorable trends with effect sizes approaching 0.2 threshold for clinical relevance. These findings provide valuable insights into the trial results with further endpoint analyses ongoing. Importantly, no cases of ARIA occurred during the trial, despite the inclusion of participants with known risk factors for ARIA. In fact, approximately 35% of the total population had an increased risk of ARIA. XPro was found to be safe and well tolerated in early Alzheimer's patients with biomarkers of inflammation, multiple comorbidities, and a median age of around 74. The most common adverse event was injection site reactions, which were generally mild and occurred more frequently within the first 8 weeks of treatment. While most injection site reactions were manageable, 20 patients required dose modifications and 10 discontinued due to injection site reactions. An additional 4 patients withdrew for unrelated reasons. Importantly, injection site reactions were not unblinding and were more of a nuisance than a significant safety concern. Other common adverse events included headache, arthralgia, and upper respiratory infections, with no significant differences between groups. Serious adverse events occurred in 6% of patients with no group differences, and no deaths were reported during the study. Importantly, there were no emergency evaluations or unscheduled neuroimaging for the neurologic events such as stroke, seizure, or visual disturbances. While XPro did not meet the primary endpoint in the MITT population, a favorable benefit of XPro over placebo was observed in a pre-specified subgroup for which XPro was expected to benefit the most. These are amyloid-positive patients with high levels of inflammation. In this group, cognitive and behavioral benefits were observed that are both consistent with the mechanism of the drug and relevant to the trial design. A favorable impact of XPro was not observed on functional measures, which may be attributable to the duration of the study. Finally, XPro is safe. There were no cases of ARIA, and the most common adverse event was injection site reactions, most of which were mild or moderate. Finally, I'd like to thank the patients, caregivers, and sites for their participation in this study. We are excited to share additional data in time as we look forward to advancing this promising treatment forward.